the title of my presentation sounded different on the paper. As I was preparing to it, I decided to rephrase it. And this is a question: reproducibility crisis, a God-sent opportunity for STM publishers? At the end of the presentation, I want you all answer yes to this question. Okay, great. So it's myself somewhere in 2004 at Princeton doing my PhD. You see less gray hair, less pounds, better looking. And my boss, my supervisor, is asking me to reproduce an experiment. And again, what is happening here? Great. So this is an experiment that was published a few years before that in one of the most prestigious journals, and it was about a new way to grow embryonic stem cells in the laboratory. So he is asking me to repeat it. And why is he asking me that? Because in biology, in biomedical sciences, this is the only way to learn new technology. Let's say you're at the lab at Princeton, someone at Harvard developed new technology and published about it in a journal. The only way for you to learn this new technology is by repeating it with your own hands. And this is why scientists, most of the time, actually busy in repeating work of other people trying to learn that new technology. So I take the journal, I get to the methods section, I read this technical mumbo jumbo, which is very characteristic to every scientific journal, and I'm trying to obtain result. And the result looks like this. Basically, your cells growing under microscope. It didn't work. As oftentimes, it didn't work. And then I tried to do it again and again and again. So let's define reproducibility. It's ability to reproduce a published experiment. And my experience was anecdotal, and there were many anecdotes like that around me. Well, it's difficult. Biology is difficult. Biomedical science is difficult. That's why we are doing PhD here. That's why we are suffering. But since 2012, anecdote was supported by data. There were two companies. One of them is German Bayer, which took 67 projects in published articles in oncology, women's health, cardiovascular medicine, and put the 100 scientists on this project and asked them to repeat the findings there. They were successful, replicating only 21% of published studies. Then another company, Amgen, in California, repeated the same thing. They took lots of their scientists, put them on 53 landmark articles, meaning articles published in good scientific journals with good significant results, and they tried to reproduce them. Their results was even worse. They got only 11% of studies replicated. And there was an academic project in psychology where they took several psychology articles and tried to reproduce them. They got to 33% of replication rates. In any case, the 60 to 90% of published science articles are not reproducible. And this is a hair-raising number, because that means that most of the science which is published today, it's not useful. And why it is a problem? Because it damages the science. So let's look at the cycle of academic science, academic research as it's going on. So as a scientist, what you do is you do new research. And once you obtain results, you publish them. And when you publish them, other scientists in this almost technology transfer process are supposed to read and learn that published technology. Well, when that doesn't work, the entire thing doesn't work. No reproducibility, no knowledge transfer means no productive research. So let's translate this problem into numbers. So in US only National Institute of Health, one of the major funding bodies, contributes around $40 billion per year to fund biomedical research. If 60 to 90% of this non-reproducible, means up to $36 billion per year go down the drain. If you expand this problem across the entire world, that number probably goes 5-fold higher. And that's the cost of the problem. And the cost is huge. Cost is huge at every level. For the society, it delays in the development of new medicine and low ROI, return on investment. This is all taxpayer money. It also undermines fundamental belief of the modern society in science, because science, by definition, is supposed to be reproducible. If it's not reproducible, it means it's not science anymore. For institutions, it means poor productivity. For scientists, it means broken careers. How many of us come to science with ideas of curing cancer, developing new stem cell therapy, or new drug against Alzheimer? What we end up doing is to trying to repeat already published experiments again and again and again. So how do scientists typically deal in real life with this reproducibility problem? So this is an example of another study. The original study was published in Science in 2010, and it showed an effect of specific chemical on tumor. You can see that first row where you see the effect. And then a replication study was recently published in eLife when they were not able to reproduce this study. So in such situation, the few choices given to scientists are this. One is to try to do this experiment again and again and again. Maybe you missed something in that text description. Maybe you don't know exactly how to shake your cells before you put them into incubator. Maybe authors did not mention that the cells supposed to go to freezing before the experiment, or whatnot. So you repeat the experiment. It's a multi-stage process. You try to manipulate each of the stages until the experiment works. It takes weeks, sometimes months. I met people who spent half of their PhD, a few years, trying to reproduce a single experiment, trying to learn that technology. Another solution, find someone at your institution who can show you the experiment. Sometimes you are lucky. Sometimes you find someone, hey, this guy did it before at their previous institution, and they know how to do it. You come to them, you are nice to them, you're asking them, can you show me? They show you. Once you're shown the experiment, you can see all these details that you cannot visualize from reading the text. Then you can repeat the experiment. But most often than not, such help is not available at your institution. Why? Because science is very narrowly fragmented. Neuroscientists and biochemists use completely different methods, so they cannot help each other often. Third one, travel to original authors who can show you the experiment. If you are lucky enough and you know those authors personally and you have grant money to travel, you go there and they show you how to do the experiment. And again, you know everything. And that happened to me in my career where I was traveling from Princeton, New Jersey to Edinburgh, UK to see one experiment. So that shows you extent to what people go in order to reproduce an experiment already published, already in prestigious journal. So if you notice, most of the time, this problem is solved by show. So why does show work? What does show provide to a scientist that text article cannot provide? So let's see it based on this example. So this is another experiment. Again, very typical descriptions that you can find in many science journals. And it shows an experiment on oocytes. So let's try to read an experiment just for an exercise. Position the metaphase spindle at 3 o'clock and hold it with holding pipette. Apply piezo pulses to penetrate zona pellucida, etc., etc., etc. Who of you understand this? Raise your hand. You do? Great. Who else? Okay. Can you repeat it in the lab? Done it? Okay. All right. One, you know, I have a PhD in this area of research. If I would get it first time, I wouldn't be able to do it. And so, but how can we switch on this video? Yeah. Let's look how it looks in real life. That's all. And even if you don't know anything about stem cell research, oocytes and nucleation of oocytes and all these technical terms, you understood what happened here. There was a ball, there was sticks that came to the ball, scooped something, that's all. That's the end of it. That's why video is so powerful in learning the methods. So all this problem of reproducibility, it's mostly because of methods. It's today, scientific article is very inefficient to transfer this methodology, to transfer this how-to knowledge, right? Video can do it. So, and again, about the today format of science article. If you think about science, right? Science has progressing very fast. It changed dramatically during the last 100 years. The scope and depth of our knowledge incomparable to what we knew 100 years ago. It's definitely incomparable to what we knew 400 years ago. But when you look at the format of science article itself, it didn't change that much. So this is an article published in 1665 by Royal Society. This is an article published by me in 2006 in PNAS. Tell me, what's the difference? Colors, we have color figures, and now searchable online, but the core format of science article has not changed. It's almost the same. And, okay, so while we have this problem of method transfer, of how-to transfer, we at the same time, since 2012, since the problem of reproducibility became obvious, you hear people and organizations come with all kinds of solutions. One of them, more data preservation. We publish articles. Scientific studies typically generate a lot of data. It can be sequence data, it can be microarray data, it can be other data. We should make sure it is well preserved. Another one, better training in statistics. Scientists don't know when they do measurements what is statistically significant, meaning they are not trained enough in statistics. So let's train them statistics. Another one, today scientists don't have incentives to publish those studies, to ensure reproducibility of those studies. Let's apply all kinds of administrative measures against them so that they will publish research reproducible. I think that these methods do not improve knowledge transfer of methods, the main problem. And I think that they will not solve reproducibility crisis. What will solve reproducibility crisis is to improving the core issue, which is the today format of the science article. We need to make sure that science article format is able to do what scientists do in real life, meaning they show experiments. And why, for example, statistics training wouldn't help? Again, coming back to the example I showed you before, this is an article in Science. You see the result. The result shows 7-fold difference between the result and control. When you get 7-fold difference, you don't need any statistics. You can repeat your experiment twice. If it shows the same result, done. This is a true result. The reason this result cannot be reproduced oftentimes, most time, is because someone who is trying to reproduce it does not know how to achieve the result, does not know the method. And statistic training has nothing to do with it. So it's all about method. So I claim that STM industry can solve the reproducibility crisis by improving the knowledge transfer of how-to, knowledge transfer of methods. And I'm going to show you 2 examples. One is a visualization, what I showed you before, how Show Me works in real life and how it can be done in scientific article. And it works on the example of our job in biological and physical sciences. And then I will show another example from Elsevier, what they did for their experiment in computer science, introducing interactivity features. So let's start with visualization. 10 years ago, after encountering those problems in the lab, we started Jove. What Jove does is film experiments in universities around the world. Then, based on those, we edit these films, we publish them as scientific articles together with text. This material is peer-reviewed. While the format of these articles is new, we at the same time use traditional processes of scientific publishing industry. We have peer review, editorial board, journals indexed in Medline and PubMed. So it's a combination of new format and traditional processes. One of the, as we started this project, one big question was, what would be the format of video article? No one has published video article before. So we decided to copy the format of the traditional text article where you have abstract, introduction, experimental procedure, and discussion of results. And so all our videos are structured in this way. So let me tell you now about the adoption. I told you the project, which then became company, started 10 years ago. As you see, we received more and more attention. At this time, we publish more than 1,000 video articles every year, about 100 per month. They're all filmed in different universities around the world in about 25 countries. So far, we have about 9 million of users per year, and 80% of traffic comes from academic institutions. On the business side, we use subscription model. So most of our subscribers are top research universities. At this time, about 1,000 research and academic institutions subscribe to Jove, and the usage is high. Every video is viewed more than 2,500 times per year. Okay, so finishing that visualization and adoption of visualization, let's move to the next example, interactivity. So when you do computational studies, either in computer science or in bioinformatics, when you read an article, oftentimes you would like to run the same computer code, and you would like to get the same results that they describe in that article. Well, typical format does not allow you to do that. You're supposed to read it, then go and code it, and if you're lucky to find a piece of the code, maybe you will integrate it somehow into your program, and hopefully it will work. So a few years ago, Elsevier came with the project of executable paper, where they embedded code right into the article. And that embedding allows manipulation of the parameters in the article, input parameters, and you actually can run the program in the article and see the results. So again, they integrated executable code and data into the article. It allows to run the code and also visualize the result. And this is another, I think, example of the good example of change in the format of the article that enables you to increase reproducibility. So what does it take? And I put these slides as no pain, no gain, based on our own experience with changing the format of scientific article. So first of all, it's more expensive. So in the traditional workflow of scientific publisher, you are in the office, you get an article submitted, you send it to peer review, it comes back, you decide on publication, you publish, end of it. Now you can sell it, right? It's the end of your work. When you deal with new formats, that's only the beginning of the work, right? In case of Jove, we get the article, we run it through peer review, and now we have to go to the long process of video production. And it's one thing when you produce one video per month, totally different thing when you produce 100 of them per month. So cost. Totally different requirements for technical expertise. So you need, in addition to all the traditional people, editors and such, you also need video editors, and you need programmers, and you need to assemble it all online in the good, readable, digestible format. So as I said, it requires big changes in traditional process. And it requires significant upfront investment for doing all this. Author pay model, oftentimes called open access model, is not applicable here. Why? Because cost per article becomes expensive, high, right? Authors today are ready to pay. If you have an open access journal, your authors would pay $1,000, $2,000, $3,000, $4,000, not more than that most of the time. Here, the cost per article becomes high. Authors wouldn't pay $10,000 per article. But what if that's what it takes to produce your article, right? Which brings us to the conclusion, it is an opportunity for STM publishers. Because the traditional publishing with introduction of internet became cheap. Right? Today, everyone can start a website, very much as you start a blog. You can call it a journal. You can call a few friends to become your editorial board, and you're good to start a scientific journal. Very different from the print area. This technology requirement brings us, in a sense, back. You cannot do any more on your knee overnight to start a scientific journal. It requires much more investment. It requires much more expertise, something that not everybody can do. And let's look again more reasons why I think the reproducibility crisis, or need to deal with this reproducibility crisis, brings an opportunity for today STM publishers. So current STM issues. One is low barrier to entry, as I told you. Yes, traditional journals have much higher quality requirement, and they are much better at ensuring the quality through peer review processes and so on. But significance of this is becoming less obvious for users, or at least some of the users, right? So that generates a negative perceptions among some users and very vocal users and some buyers. And that brings us government mandates that challenge the traditional models. Well, improving article format will increase barrier to entry. It also will improve user perception, because nobody will be able to claim anymore that publishers do nothing. Here it's very clear, when you do video, you do video. It's not author creates that video. When you introduce interactive features into your articles, you introduce them. It's all done by you. Yes, it's more expensive, but it's also more useful. And I think in the end, it will strengthen the traditional subscription model. Well, with that, it's my question to you. What is your move to deal with the reproducibility crisis? Are you going to publish this or that? Thank you.